So what we're going to do today is talk about these two papers, and then if we get through them, a little bit of material, as you will see from previous lectures, that relate to what you do with ribosomal RNA PCR data beyond just characterizing a particular sample. And these two papers are really about using phylogenetic analysis of ribosomal RNA PCR data to ask what kinds of organisms have we characterized via culturing, and are there any differences with the kinds of organisms that we see in environmental samples? That is, are there groups of organisms that we know nothing about because they've never been able to be grown in the laboratory? And this is a large component of what ribosomal RNA surveys have been used for. Even to this day, people are still finding with ribosomal RNA PCR new phyla even. I didn't want to go into the latest papers, but in talks by Norm Pace, who's on both of these papers here, his current analysis suggests there are something on the order of 200 phyla of bacteria, of which, they talk about, 20 or so of them in these papers. And we still have not really sampled diversity even with those 200 phyla. But it's kind of completely overwhelming to deal with that data. And these two papers are the most sort of comprehensive and heavily cited and sort of detailed about how people use ribosomal RNA PCR data to look at novel taxa. And then in the next class, we're going to go to the next step, which is to say, how can we use ribosomal RNA surveys to ask questions about ecological diversity of organisms? Something called richness, which is how many species are there in a sample. Compare the diversity between samples, much like people do with plants and animals. Estimate the relative abundance of taxa and other things that you can imagine would be related to ecological studies. So what I've done here, and hopefully we can at least, even though I haven't given you questions in advance, at least pull out of some of you some comments about these papers, is basically I've done what probably most scientists do when they first look at a paper, and I assume many of you sort of do the same thing, which is to look at the pictures, to look at the figures. A good paper, the main points in the paper will be presented both in the text and visually, and frequently the visual presentations are actually better than the text discussion. Now, this is not always the case, but I picked these two papers in part because they had somewhat interesting and useful visual presentations of data that kind of summarize most of the main points that are found in the papers. So does anybody want to take a crack on explaining what is some of the things represented in this first figure from the Hugenholz paper? Yeah.
Well, they were reconstructing, a phylogeny of bacteria and whether the groups had cultured or uncultured members. And I was a little bit confused about the size of the blades, but I think that was how many representatives they had found.
Right. So the size of these little parallelogram triangle objects is an attempt to represent the phylogenetic diversity in that group. That is the breadth of evolutionary diversity in those groups. What are the colors? What does the shading versus not shading represent?
I think shaded is culture.
Yeah. So the branches where there's dark shading are the branches that include at least one cultured representative. And the branches where there's just white are those lineages that at the time they drew this tree and did this paper, there were no organisms that had ever been cultured in that group. Another way of viewing this is all of the sequences that were mapped to that group came from environmental samples, and none came from cultured organisms. At the time they did this, so they go into a little bit of detail in the methods. I sort of been leading up into, what masking is and what these phylogeny methods are, and he sort of talks about this. But an important component of this is where they are, and they mention this in here, where they're getting the data to then analyze. So did people see where they're getting this data that they build the tree out of? Yeah, so there was this database. It turns out it is still one of the heavily used ribosomal RNA databases called ARB, ARB, which once ribosomal RNA sequencing started taking off, you started to see the proliferation of different databases where people would store that information, make it available to other people, and make certain types of analyses of that data available to other people. And so I don't know if people are familiar with this, but in most scientific papers where people generated sequence data, after a few years, people started to generate this data. There was the creation of a couple of databases where people would share that data. And pretty soon after that, journals started making it a requirement, if you published a paper analyzing sequence data, to make that data available in one of these databases. And the sort of biggest one still now is called GenBank. It's run by the United States NIH. There's a group called the National Center for Biotechnology Information, NCBI, that maintains this GenBank database. There was a big database in Japan that a lot of people used. It's still there, but not as many people use it anymore, called DDBJ, I don't know what it stands for actually, D something, DNA database of Japan. And then the European Union created a database that is now, I think EBI is what it stands for. And they're sort of spiraling. Sometimes they grow, sometimes they shrink. But the main ones people use now are GenBank and EBI. And what people found was those databases were really just places to dump information. And it was not easy to pull out information that you were interested in out of those databases. And what's called annotation of the information. So if you had a sequence in there, information about where the sequence came from, how the sequence was generated, alignments of the sequences, those were not in GenBank or the DDBJ or the European database. And so a bunch of people who focused on ribosomal RNA analysis started to create ribosomal RNA specific databases, which people started to use. And one of them was ARB. And it's still used extensively. And for example, you can go to these databases and select now a taxonomic group. You could download all of the sequences from bacteria, or all of the sequences from Escherichia. And you can obtain sort of environmental information about where the sequences came from. And probably most important, you can get an alignment of sequences, a pre-existing alignment of sequences. So if you wanted to analyze, I mean, even here in this paper, they probably analyzed a few hundred to 1,000 sequences. Making a new alignment with 1,000 sequences is not trivial. So it would be better if you could get the alignment that other people made a year before and then reanalyze that. So that's basically what they do, and what people still do in most papers is they download a database of pre-existing ribosomal RNA alignments and then analyze their new sequences relative to that. Some of these databases are more or less useful than others. After using ARB for many years, the lead author on this paper, Phil Hugenholz, then created his own database called Green Genes, which was much more curated than the other databases. So they threw away crappy data, basically, because many of these databases accumulate good data and sometimes bad data, and they tried to be really stringent about what data you used. And anyway, it's just when you get into analysis of any of these new types of data, for any field, there's probably going to be a large repository where everybody dumps everything, and then what we kind of call boutique databases, where the cleaner, prettier, curated information is. So another thing that this shows is, you can count now the number of major lineages that are represented in this tree. So at least at some level, the number of these branches represents at the time the well-supported phyla of bacteria. They didn't include, so if there was one sequence that was sticking out on its own, seemed to be potentially novel, but you didn't have a lot of statistical support for, they may have thrown those away. So these are sort of the confident major groups of bacteria as of that time. And again, as I was mentioning, a lot of them have numbers for their names. So all of these OP were isolated from octopus pool. OS is octopus spring. TM, I don't remember what TM is. And WS, I'm not sure what WS is either. But anyway, these are, and then the other ones, generally if there was a cultured representative in a group, it would get a name based upon one of the cultured representatives from that group. And the ones that didn't have any cultured representatives did not get names.
I was kind of unclear on that figure. Is there a branching pattern, or is it just kind of like...
So I think purposefully they made it hard to see the center part of this figure. And if you look in the Pace paper, he has one figure, as we'll see in a little bit, where he just completely obliterates any information at the middle of the tree. And I mean, Hugenholz and many other people have written papers about the deeper parts of the tree and whether or not there is support for some of those deep branching patterns. I don't think they wanted to deal with that in this paper, and so you'll see that they show the total tree, and then they show within phyla, and they don't show the in-between. Probably because there wasn't a lot of statistical support, and they didn't want to argue with reviewers about whether or not those patterns were robust.
I put aquafacalis as the deep branching one.
Right, so they do mention that, and this has been a source of consternation among many people in the field. So they put this one phylum, the Aquafacalis, as the deepest branch. There are a few cultured representatives in this group. Most of them had been poorly understood when they did this. One is called Aquafex, that's the genus. Another is called Hydrogenobacter. And there was not a lot of evidence for that deep branching pattern of Aquafacalis. Other papers around the same time had the Thermatoga group, Thermatogales, as the deepest branches, and there were a whole series of papers based upon this ribosomal RNA tree pattern because the Aquafacalis, Thermodisulfobacterium, Thermatogales, Coprothermobacter, and Dictyoglomus were all thermophilic. They all grew, the members of these lineages all grew at high temperatures. And so based upon that, people concluded that the most likely common ancestor of all bacteria was also thermophilic. But a lot of that is dependent upon the exact order of branching here. And we didn't talk about this, but in the Pace paper in particular, the second paper here, And in many of the ribosomal RNA studies, they talk about how great a phylogenetic marker ribosomal RNA is, which it is true that it is very useful. But it has a few limitations, and one of the limitations is massive. So when people first started to characterize organisms that grew at high temperatures, they had this expectation about what their genomes would look like that turned out to be wrong. So GC base pairs have 3 hydrogen bonds. AT base pairs have 2 hydrogen bonds. And many people expected that organisms that grew at high temperatures would have more GC base pairs in their genome in order to hold their DNA together to withstand the denaturing that would happen at the high temperatures where they grew. And it turns out this was not true. The organisms that live at high temperatures have other adaptations that help hold their DNA together, including proteins that bind to the DNA and other modifications, except for non-coding RNAs that have to function as independent RNA molecules separate from the DNA and fold over on themselves and form base pair structures on themselves. In thermophiles, all the non-coding RNAs have higher GC content. And what this means is that unrelated thermophiles, if you have 2 organisms that go from growing at low temperatures to growing at high temperatures, their ribosomal RNA sequences will actually converge upon similar GC content. And it misleads phylogenetic trees when you build them. The methods don't correct for this perfectly. And so many of these patterns in this tree are probably incorrect due to long branch attraction and convergent evolution of GC content. And protein coding genes, such as the ones used in the papers we talked about last time, are less prone to this problem than ribosomal RNA genes are. And so the deep branches in ribosomal RNA trees are particularly problematic for some.
So with GC content, right, you need it to tell horizontal gene transfer, right? So what influences it? Like, is there any sort of pattern in how it is spread across, or is it just random?
Well, so I think you're referring to how some people look at genomes and find regions that are different in the GC content than the rest of the genome.
Use it to infer possible horizontal.
And use it to basically say that something is anomalous, and the normal model for what is anomalous by most people is lateral gene transfer. Unfortunately for them, convergent evolution can also create anomalous GC content, as can weird mutation processes, and not always lateral gene transfer. And so there's actually a lot of work on what different forces can affect the GC content in organisms. It's not very well understood why E. coli has, X GC X percentage on average of its genome GC content, and for example, Plasmodium falciparum, the causative agent of malaria, has a 25% GC content, and some halophilic archaea have a 70% GC content, and it's not really well understood why. But what is clear is that GC content changes pretty rapidly among organisms, and it is similar in many ways to any other feature that changes rapidly. It can be useful for looking at closely related organisms, but not very useful for looking at distantly related organisms. Does that make sense? So I don't really know if at the time Phil Hugenholz believed the deep branches here, but they were not very well statistically supported. And then subsequent papers came out and said many of these thermophiles are incorrectly grouped together in the tree. And the same is true for the archaeal tree, and even a little bit for the eukaryotic tree, although there aren't quite as many thermophiles in the eukaryotic tree, so they don't have the same problem, the same extent to the problem. Does that basically make sense? Are there any other questions about this? This figure was subsequently used in probably 10,000 talks and 17 textbooks, and this paper was very, very influential. I don't know if you can see, I don't think I can get a better resolution figure here. Do anybody have any comments or questions about what is presented here? So this is a very different piece of information. Yeah.
Sort of the equivalent of showing the range, I guess, of different bilags in the bacterial domain.
Yeah, so what they basically did was they went to that database and said, let's look up not only the sequence of an organism, but some information about where that either organism came from if it was cultured, or where the sequence came from if it was an environmental sample. And now let's build a little table that shows it's really a niche. They're not showing physical space, but they're showing sort of the niches for different groups. And also important is that they also show the number of sequences that were in that group, which tells you a little bit about how robust this particular pattern is. So, they go through geothermal, soil, marine, freshwater, wastewater, pollutant associated. I'm not really sure exactly what that means. Acid metal leaching. I can't even tell what this one is. Sulfur. And then symbionts. And which lineages are, these from. And I'm not sure this is comprehensive enough or precise enough to, tell you anything about these particular organisms. But I think the point here is that it doesn't appear to be completely randomly distributed across environments which organisms are coming from those environments. And you're having, some groups that appear to be only from thermophilic environments, and some groups that are, soil associated. I think the acidobacterium, for example, this dark circle, if a lot of the samples came from there, is, a lot of them came from soil. So for many years, people talked about acidobacterium as a soil type of organism, and so on. I'm not sure I would make any significant conclusions now based upon this, because we have so much more data now to map the sort of distribution patterns of organisms and their ranges. But this was basically hinting at that you could combine phylogenetic information with ecological niche information and build sort of the equivalent of what you might do with plants and animals with microbes from the ribosomal RNA survey data. As an aside, you may not have noticed this unless you sort of went to the databases to look at the paper itself. There were a few mistakes in this table. And what people frequently do when they make an honest mistake or a simple mistake is they publish what's called an erratum to their paper. So the journal where you publish the paper is supposed to publish this little thing called an erratum. They're supposed to notify the journal literature databases that there has been an update to the paper, and they're supposed to sort of backlink them all to each other. It's not always easy to find this. But I just thought it would be worth pointing out that papers come out frequently with simple mistakes in them, major mistakes in them, or complete fraud in them. And the paper doesn't always stay the same over time. So they said that column 1, table 1 should appear as shown below, and then they show this much nicer table. They've compressed it so it's easier to see on one page. And they've added these check marks now to the table, and it's just much cleaner presentation of the table. And they say, I don't think they said this in the original one. Oh yeah, they do say an expanded version of the table is over here at this website, which doesn't exist anymore, of course. anyway, it's not that substantially not that different, but it's always useful to remember that sometimes mistakes are made in papers and they get updated. And if you go to like PubMed to do a search for a paper, it should say, even a paper from the 1960s will say, in 1972, there was an erratum published, see this link. Not always easy to find. There's actually a whole system now and a website to track what are called retractions of papers. So if you make more than a simple honest mistake in a paper, if you either invent something or you can't reproduce something or there's some major problems, people will actually formally retract the paper. Those don't get tracked very well either necessarily. And so now someone has created a whole database basically to help people track the retractions of papers. Anybody want to take a crack at explaining this figure? All right.
So it's basically you have the different sort of major groups of bacteria, and they're showing sort of the percentage of the known, I guess, species of them that they have cultured representatives versus non-cultured. And you can sort of see the ones that there are as much culturing work to be done.
Yeah, although the way they get the numbers here is they're not showing total numbers, right? They're showing percentages. So, for example, OP11, I think there were like 4 sequences, maybe even 3 sequences at the time. So it looks like, when you look at this, like, oh my God, there's so much uncultured diversity. But really, there's just, a couple of sequences and no one has grown anything from that group.
There probably isn't cultured diversity.
So many people criticized this figure for many years because of that issue. But I mean, I think that this was really important to the community. It's sort of the zoomed in version of this, saying, let's look within the phyla rather than between the phyla. Sure, some phyla have no cultured representatives, but let's look within these other phyla and ask what fraction of them have been cultured. So for example, Acidobacterium, I don't remember if you look at the table. No, it doesn't say in here. Acidobacterium, there probably were, 30 clones or something like that, and one of them had been cultured. And Verruca microbia, I think it was, 40 clones, and 2 of them had been, 2 organisms had cultured. And so this is, when you see data like this, you should think about where all of the information comes from. So, for example, at the time, and even now, there's a collection of cultured bacteria and archaea. At the time, most of even the cultured bacteria and archaea had no ribosomal RNA sequence associated with it. So all they could analyze here were the ones that someone had sequenced a ribosomal RNA gene from. So there may have been many organisms that people said were in the actinobacteria, for example, but the only ones they analyzed were the ones that had a sequence available. And so this gives you a sort of incomplete perspective, both because it's not showing total numbers in any of the parts of the figure, and it's also not telling you about all the other organisms in the group that just didn't yet have ribosomal RNA sequences available. Even today, there's an effort by the German culture collection, the DSMZ, and there's a big US culture collection called the ATCC, to finally obtain ribosomal RNA sequences for everything in their collection. So even though people can now sequence 200 billion ribosomal RNA sequences from the human microbiome, and there are only 10,000 formally described species of bacteria and archaea, not all of them have a ribosomal RNA sequence available from them. So it's just this weird sort of historical artifact that people have cultured organisms, they typed them by some other information like DNA hybridization, which was commonly used, and they never obtained a ribosomal RNA sequence, and they're just sitting there somewhere, and people haven't yet gotten back to characterizing them. And then, I don't know how much it's worth going into the details on all of this, but, I find a figure like this sort of incomplete, and a figure like this incomplete, and what they were trying to do here is show you the full breadth of some of these phyla. So here's the Acidobacterium group, and that's what I was saying. There's one cultured species shown in bold in the Acidobacterium group, and all the other representatives of this group are represented just by clones. I think these are the outgroup, Geothrix and Halophaga. I don't think they're in the Acidobacterium division, but I'm actually not 100% sure. Maybe they were. So maybe there were two other organisms that mapped to the Acidobacterium group. I don't know if they still are in that group. But anyway, this is a much better way at looking at the diversity, because you can see not only are there many sequences from environmental samples, they're really diverse. That is, there's a whole clade here, clade 3, and a whole clade here, 4, and 5, and 6, and 2. Subfamilies or, orders of Acidobacterium that had no cultured representatives. And then, Verruca microbium, there were 3 here that were listed as having a name and cultured representatives. And again, there are multiple lineages within the group that had no cultured representatives. And same thing for the green non-sulfur group. There was one clade that had lots and lots of cultured representatives. Many of these were cultured in part because they're photosynthetic or they're phototrophic in some way, so people were interested in the members of these groups and spent a little more time trying to figure out how to grow them in the lab. And many of these other ones, people would see them in some environment, but they didn't have, like photopigments, or they didn't have as much of interest that people didn't go out of their way to try and figure out how to grow them. But even so, this is just basically the whole point of these 3 figures is to show how poorly sampled the culture collections are, how poor the culture collections represent diversity even within these phyla. And then this is just showing that OP11 has a lot of hidden diversity, and none of them have been cultured. So there was a paper that they wrote before this that was just on Yellowstone hot springs that showed the same thing, and then this is sort of the let's show it across all environments. But these 2 papers, the Yellowstone hot spring paper and this paper, basically convinced a huge fraction of the community that it's not just sequencing, 16S from environmental samples is basically a window into the vast representation of the diversity of microbes that had not been characterized at all by culture methods. And then the PACE paper is basically a much more detailed analysis of the same argument. And he goes into a lot more justification of the methods that they are using, as well as trying to teach people. So I think his point here was to review the state of the art of what ribosomal RNA surveys revealed, 10 years after the previous paper, or 11 years or so after the previous paper, and also to reiterate some points about the methods that people use and or shouldn't use, and how they work. And so, did anybody understand this figure?
Wasn't it only see so much change in the sequence because some of the substitutions that happen reverted to the ancestral sequence. So there's actually a lot more evolution that's occurred that you can see based on sequence comparison. So what you can see is the dotted line, there's more unseen change and better solid line.
Yeah, so that's exactly right. So as time goes by and you're comparing any feature of organisms, what you have happen is that you can get a change in that feature and then a change back to the ancestral. So you can change from an ancestral to a derived state and then a derived to an ancestral state. And if that happens, you would mistakenly underestimate the number of changes, the differences between organisms because of this hidden change in the data. And there's a simple mathematical formula to correct for this that has been used for many, many years in any type of comparative data, and that's the formula shown in this in this figure here. And it's basically pretty straightforward. You ask how many character states are there. So in DNA, you have 4 character states. And so if you have, if your starting sequence is just a run of A's, those A's can change, each one of them can change, either stay the same or change to C, T, or G, right? And then you continue to have evolution, and if, it changed to a C and then changed to a G, and then eventually changed back to an A, and you're comparing to other things where there was no change, you would mistakenly underestimate the number of evolutionary events that have happened. The more character states you have, the less of a problem this is. So if you have 1,000 different possible characters that each DNA base could come in, the probability that you would go back to the original one just randomly is much lower. The fewer character states you have, the more of a problem this is. And so this formula allows you to calculate the expected, the amount of change correcting for what you observe. Yeah?
If there was 1,000, like even if there was more character states, couldn't you still miss a lot of changes? Because there are changes to the C and then the G. If you just added the G, you would miss that other evolutionary step.
Right. So if you imagine an evolutionary tree and you have an ancestral sequence, you can have changes anywhere along this evolutionary tree. And if you had a change, let's say here, at the first position from A to C, the good thing is you wouldn't have had a change here. And as long as there wasn't any other change along here, you could use that to figure out that this ancestor was A, and there must have been a change from A to C. And so the better sampling you have, the more likely you are to be able to figure out that there was an A to a C to a G change. But without any of that information, if you're doing what they're doing here, which is just calculating a distance between sequences and not using the tree, you're exactly correct. So once you change from A to C and then C to G, it's also you lose the information of the A to C change. And so what this formula basically does is converts the distance that you observe to a corrected distance. And the further apart, the more dissimilar your sequences are, the more correction you have to do because of this exact issue that you just mentioned. So, and again, the fewer character states that you have, the more correction you have to do also. So it's the combination of how dissimilar the sequences are. That's why the number of changes is in this formula, the D here, and then the 3/4 and 4/3 is basically the total number of character states over the number of character states minus 1. So if you had amino acids, it would be 20 over 19. And this is a standard formula used to convert an observed similarity score to an evolutionary distance measure. And he goes into this a lot because many people published lots of papers where they didn't do this. And they would take the ribosomal RNA sequences, align them, calculate a similarity score, and directly build a tree from that similarity score. And that's fine for the tips of the tree because the observed distance is not that different from the real distance when you're looking at similar sequences. But for the deep branches in the tree, people wrote a lot of papers with really weird stuff in the deep branches of the tree when they didn't make this correction. It's still hard when you make the correction, but it's a lot worse when you don't make the correction. Yeah.
So then I guess they're assuming in this formula that the changes happen randomly? Or like in some sort of statistically...
This is just a statistical, Poisson expectation, I think, of random change.
Yeah.
And so now imagine that change is not random. For example, unrelated organisms get selected for high GC content due to their growth at high temperatures. You would actually still mistakenly calculate an evolutionary distance in that case, and unless you had a model that incorporated growth temperature into your calculation of the distance and corrected for that. And so in the papers, we skimmed over the model details. In the papers looking at the eocyte tree and the 3-domain tree, the models that they use are very complicated. Amino acid substitution models that take into account things like the chemical similarity of amino acids, and if the ancestor had an overrepresentation of alanine, the probability that you see another alanine might be higher because there may have been just sort of lots of alanine in all their proteins, so they try and account for that. We're sort of glossing over all of those details. But good evolutionary reconstruction methods don't have to do this. This is specifically for these distance methods. Because once you calculate a distance and feed it into your phylogenetic tree algorithm, you lose all the data in the sequences. Likelihood and Bayesian methods and parsimony methods retain the actual sequence data, and you can have a model that says a change from an A to a T is a higher probability than a change from an A to a G. Transitions versus transversions, for example, or something like that. So I think his point here was just to emphasize that you don't just take your data and throw it into a clustering algorithm that you pulled off the shelf. You have to think about a model of evolution in order for this to work. Yeah, go on.
Well, David's always talking about, he gets upset when you look at just like the percent identity match and not looking at the tree because something can be more similar, but it's not actually the sister.
Right, so on top of all of this, there's a separate issue, which is that if you imagine an evolutionary tree where the branch length in the tree represents the amount of change, then if you calculated percent similarity among these organisms or genes, that would be represented basically by the total length that you have to traverse from the different organisms to each other. And in this tree, the highest percent similarity is going to be between B and D if this branch is really long. And if you used an evolutionary method that first just grouped together the things that had the highest percent similarity, they would artificially group B and D together and get that incorrect. And so this
is long branch retraction?
This is not long branch retraction. This is unequal rates of evolution. And so what happens is that, and this is partly the point of what he's saying here, what has happened in the field has been that a lot of people are very used to taking data that they observe, like shapes of furniture or planet shapes, and clustering them so that you can group the objects that are most similar to each other. And if you ran one of those clustering algorithms on data with a history, they will group together the things that are most similar. And so there's a method that many people still use for building evolutionary trees called UPGMA that will get this wrong every time. And that's why I was hinting at this before. For distance methods, many people switch to neighbor joining because neighbor joining allows the length of branches to be different and will correctly build a tree from this type of data. Now, long branch attraction, well, do you have a question about this? I'll get to long branch. So long branch attraction is a separate issue, which is that imagine it's like the inverse of this issue. Imagine this is what the tree really looked like, and you got data from A, C, B, and D. And even if you ran a method that did not only use percent similarity, if there were high rates of evolution, you would have a lot of changes on these two branches because they're longer. That's what the long branch is supposed to represent. Evolutionary methods, all of them, get confused when you have two branches that have had lots of changes in them. And they pull those branches together. And then they will artificially group C and A together, all of them. Now, likelihood methods are better at dealing with this than parsimony methods, but they're still prone to this problem. Does that sort of make sense?
I don't completely get the difference between the two.
So the difference is that there's a first problem, which is that percent similarity is not a perfect indicator of evolutionary relatedness. And if you want to take data on similarity of organisms and convert it into an evolutionary tree, you have to use a method that allows for rates of evolution to differ between different branches. And that will correct for this problem of percent similarity being misleading. And what they do is what's called a relative rate test. It's very simple. You basically ask, does the distance from B to D equal the distance from B to C? If rates were all equal, then the distance from B to C should be the same as the distance from B to D. But if they're unequal, you can see that the distance is skewed. And that's what most of the methods do to correct for this. Long branch attraction is a similar issue, but it's not based upon using similarity information. It's based upon even trying to analyze the actual character data, like the A's, C's, T's, and G's. And even if you used a model that allowed for branches to be different lengths, that would correct for this problem, these long branches still get pulled together because many changes have happened on those branches. So if I drew a tree like this, and I used a method that relied upon similarity alone, it would group A, B, and D together. But there's no long branch attraction here because there's only one branch that's long. It's artificially grouping together things just because they're most similar to each other. If I added into this tree a really long branch for B and C, now B and C are going to get pulled together by most phylogenetic methods. Does that now? So they're related to each other, but they're not the same statistical problem.
So that's because in the one that you're going to have a quiz on, it says that improved sampling can have positive effects on phylogenetic reconstruction because it helps break up long branch attraction.
Yep.
So does that mean because you're putting, now that you have more diversity, there's more space between your possible long branches?
Yep.
Okay.
So imagine if you had, instead of these, let's say the real tree looks something like this.
Okay.
And if you first sampled any of the representatives of the B group, let's say you sampled this one, B2, and you sampled C3, you would have a long branch leading up to C3 and a long branch leading up to B2, and you wouldn't have any of these organisms sampled yet. And if you start adding in the samples, you now break up these branches so that allows you to help correct, basically, for the artificial attraction of the long branches. So this is very, I don't know if anybody did this before this paper. I'm just going to show the zoomed in version of this. Did people understand what's in this presentation? So it's basically what's called an accumulation curve, now showing how many taxa you see over time. So he basically went back and took the data that you would have seen in 1989 or 1992 or 1995 and 1998 and asked how many representatives of these different groups do you see in the data? And what does this say to you? I mean, what does this result say to you?
You know a lot more about the bacterial sequences than we do about, hasn't been a lot of sampling of the archaea.
Sure. Yeah. Right. So where's the asymptote on this? There doesn't appear, at least in this figure, to be any slowing down of the accumulation of representatives of these.
Is this new sequences or just sort of
Oh, you know what? He may have included all sequences in this. I take it
It
back.
doesn't look like a refraction.
Right. So I'm sorry. That's this is just showing how much data you have. If you collapse the data, we'll see this in the next week classes papers. If you collapse the data into all closely related sequences become one point in your graph, basically a species or what we're going to call operational taxonomic units when we look at this, you still see, at least up until that point, an accumulation and not an asymptoting of the graph. Now, today, you're starting to see somewhat of a slowdown for some groups. I think I'll try and find one of the latest papers on this, but
By group, do you mean domain or group meaning bacteria?
the things I've seen are phyla.
Okay.
I don't know if I've seen it for all bacteria or all archaea. And the other thing shown in this figure is then for each of the groups, how many have been cultured, or really how many sequences are available from cultured representatives and how many sequences are available from environmental representatives. And again, there actually are lots of organisms that people have cultured, but they have not sequenced the ribosomal RNA genes from those organisms, and they have not deposited those organisms into culture collections or made the sequences available. So there very well might be a huge number of other organisms out there that have, even to this day, been cultured. But if people haven't sequenced the ribosomal RNA genes from those organisms, we have no way of linking it to this data, basically. And that is why now the culture collections are trying to go back through their entire collection. And I don't know if you know how it works. For most microbes, what they store is a frozen or dried sample of that microbe in the culture collection. And one of the reasons they avoided going and sequencing the ribosomal RNA genes for many of these is many of them are really hard to grow. So you would go back to your sample and try and grow up the organism, and you needed a shaking oil bath with weird compounds and an expert who worked with thermophiles, and many people just didn't want to deal with this. Now with cheaper sequencing and good PCR, people can go in and take a tiny little subsample without even growing it from their frozen collection of organisms and just sequence directly from that. So I think that's what the culture collections are now going to do, is to get data from all of their little vials in the freezer or freeze-dried samples. And shows this for bacteria, archaea, too. Yeah.
If they don't sequence them, how do they know that their samples haven't been contaminated?
They don't, and many of them are less pure than people originally thought. So for example, for this project I ran at the Joint Genome Institute, the culture collection would grow up organisms, and then the first pass after they grew them would be ribosomal RNA sequencing. And in many cases, we would get out 3 ribosomal RNA sequences in reasonably high abundance that were unlikely to be contaminants from the new growth. So they probably were all 3 there in the original culture. We wanted to sequence genomes of organisms, and we didn't want to deal with 3 organisms at a time. So all of those organisms got dumped. The culture collection still has a sample, but they may not be pure cultures. They may still be very interesting. Yeah, and then this gets to, let's just make the resolution of this tree even worse, and really just try and show that there are many, many branches, and occasionally there's something you can convince yourself is a relationship like, really all he shows for bacteria here are that mitochondria appear to come from the proteobacteria group and chloroplasts appear to come from the cyanobacteria group. Somehow he's convinced that there are 2 major lineages of archaea, the Euryarchaeota and the Crenarchaeota. That comes up in those papers that we just read about this is not, the best sampling, or just calling it 2 groups is maybe not the right thing, and then not a great sort of representation of all the eukaryotic diversity.
What are the unlabeled branches?
I think he didn't have space in the figure to put the names of all of them, is my guess. I don't really know.
It's funny that all of the main branching points are unresolved.
Yeah, so
That he thought
I've shown you this in a little bit of the non-historical order, which is that this came out before those papers that we read on concatenating protein sequences together to get more data. That was why those people started to say we need genome data, we need to concatenate together lots of different sequences, because the resolution of the ribosomal RNA tree is not that good. The statistical support for different regions that even appear to be somewhat resolved is not that good. The thermophile convergent evolution problem is less than ideal. So that's basically why people started saying, how can we look at other data? Basically what happened was, with those papers that we looked at, ribosomal RNA sequencing became very easy. And everybody did it for all sorts of organisms and environmental samples. And unfortunately, even though people wrote lots of papers trying to argue that ribosomal RNA was the best marker for doing phylogenetic studies, it probably isn't. It's great for the big picture, and it's cheap and easy to obtain, but it has all of these issues with it, and you basically, protein coding sequences have many advantages in a lot of cases, and whole genomes certainly have many advantages over individual ribosomal RNA genes.
So with the protein coding sequences, if you wanted to sort of that curve that we saw earlier with the differences and having more choices help, could you build sort of 3 levels of tree where you use like the codons and then the like the codons is your unit, I guess, and then the amino acids and then also
Yeah, so all, the information in organisms is basically stored in some way in the DNA, although, now there's epigenetic modifications that may be heritable and important, but let's just pretend like it's all in the DNA. You can, and people have basically found that for distantly related organisms, it is easier to compare the amino acid sequences of proteins than the DNA sequence of proteins, partly because of this issue, that is, distantly related organisms start to have multiple hits at the DNA, and you start to lose some of the signal. But a better approach would be, instead of having a 20 by 20 matrix of amino acid substitutions, would be to have a 64 by 64 matrix of codons in the protein coding region. And that would allow you to basically say that to treat the DNA in units of 3. And you could say that this, is more similar. These two are more similar to each other than these two, because those code for the same amino acid and these code for different amino acids. So it would allow you to calculate distances where you're weighting the distance by whether or not you change the amino acid or don't. And you can also then weight it by whether or not it's a big chemical change in the amino acid or a small chemical change in the amino acid. And people are now doing this. Mathematically, our computers couldn't quite keep up with the amount of data versus trying to build a tree with a 64 by 64 matrix, but people are now doing exactly that. They're called Codon Models, and people will align the protein coding genes at the amino acid level because it's easier to find the regions of homology with the amino acids, and then you back align the DNA, and you calculate the tree based upon the DNA alignment in 3 units of 3. Yeah.
How does this take into account like insertions and deletions?
It doesn't directly, but it can if you have a frame shift. So if you have a change in the protein coding region where you get a big change in the amino acid sequence, but if you can figure out where the DNA lines up and that it was really just a frame shift in the reading frame of the protein, by doing both DNA and protein level analysis, you can account for that. But in many cases, people punt on insertion-deletion differences between sequences and throw away all the data in any region of your alignment where there are ambiguities about the insertion-deletion history. Probably less than ideal, but it's probably better than using a wrong model, and we don't really know how to model it completely. Does that make sense? And Pace, I'll get back to this in a second, but Pace in his paper also talks about the issue of aligning ribosomal RNA sequences, which also come with insertion-deletion differences and other problems, and not knowing how to score it. So then another thing that he does here is similar to some parts of the Pace paper, but he now makes it prettier. To the Hugenholz paper, is mapping the sequences to the phyla instead of showing how wide that triangle is. He's now just showing you a graph of all of the sequences, and for the total data set, it's pretty clear that there are some phyla that keep showing up everywhere. People have been doing ribosomal RNA PCR, at least as of that time. So 250,000 of the sequences of something, I forget what it is, 500,000, or a huge fraction of the sequences that had been observed at this time were from one phylum, Proteobacteria. And, the top 5 or 6 phyla were a massive representation of the data. And then there's this long tail where many of the other phyla we haven't seen many sequences from those phyla. Now, what explains that? What could explain that?
Proteobacteria is common as dirt.
Sure, that could be one explanation.
The other ones can be really hard to grow in only certain areas, so it's not like there's very many niches you could go to sample.
Right, so that certainly is another one of the explanations behind this. Any other?
So we know that, we know that mitochondria came from Proteobacteria, so it's probably easier. I don't know. It might just be easier to get funding studying something more closely related to.
Yeah, well, that relates to the environments where Proteobacteria are found tend to have been more heavily studied. Probably not because of the mitochondrial issue, but because it turns out that Proteobacteria, Firmicutes, Bacteroidetes, and Actinobacteria are the 4 dominant phyla that are associated with animals. And in the human microbiome and animal microbiomes and on skin, and lo and behold, those are the environments that had been most sampled. So there's another reason, which I sort of skipped over because we skipped over one of the technical details here, at least a little bit. So with the invention of PCR, many people were going to environments using those same primers that we talked about for the cDNA synthesis, reverse transcriptase, and amplifying ribosomal RNA genes from environmental samples. And they called this universal PCR. Turns out the primers don't equally amplify all organisms. So there are lineages that have been missed and are reasonably abundant in some environments, but the PCR primers did not work well for them. And there are other lineages that, these probably aren't at the phylum level, but there certainly are families of organisms and maybe even orders where the ribosomal RNA genes are split into different fragments scattered across the genome, and the primers that people use won't amplify anything, even though they bind. For PCR to work, the 2 primers that you use have to be reasonably close together in the genome. And if the left half of the 16S is in one part of the genome and the right half is in another, PCR won't work. Yeah?
They talked about copy number as well. Does copy number of PCR genes vary a lot among these?
Copy number of ribosomal RNA genes, we will also talk about in the next few weeks, varies in bacteria and archaea between 1 and I think 18. So it's almost, a 20-fold difference. And if you're estimating relative abundance of taxa from the number of sequences you get, and you haven't corrected for the fact that some have 1 copy and some have 20 or so, you're going to get incorrect estimates. Eukaryotes are much worse. There are some eukaryotes with a couple and some with 10,000. And so when you get into using ribosomal RNA PCR to characterize eukaryotic diversity, the relative abundance issue is a much bigger problem. Yeah.
If they wanted to look more closely at some of these cultured or like the phylums that have been hard to see, couldn't they design a primer based on like a specific member of that phylum's ribosome? Because that would be more likely to be closer related.
And people have done that. It's just many people either thought these really were universal primers or didn't want to bother with designing phyla-specific primers instead of using broad primers. So on average, those groups for which the default primers don't work very well for have not been well sampled. And even if you design primers based upon the 10 nitrospira sequences, that'll only capture the diverse part of the diversity of the nitrospira. This is just showing another perspective on the same data, which is now showing the phylogenetic diversity at different time points. Instead of just the total number of sequences now, this is the phylogenetic diversity in the groups in 1993 versus 2008. It's changed a little bit, certainly within the archaea, but not a ton, at least in this figure. Oh, did I put in the wrong? Was there a bacterial version of this? I don't remember. If there was, I didn't put it in here. I don't think there was, actually. And then, sort of just at the end, he starts to talk about how there's lots of, potential in looking at eukaryotic diversity, although mostly people have ignored this because the primers haven't been optimized and eukaryotes are kind of icky. So. And then there are many other issues that are discussed in the paper. We've talked about some of them. This unseen changes. We talked about the testing the trees. We didn't talk about really at all. So there are a lot of different approaches to taking a tree and testing the results. And one of the things that he sort of talks about in here is looking at biological features of organisms and seeing how well the sort of biological features of organisms map onto some of these trees. Membrane structures or other things. Orthologs and Paralogs has come up in a couple of the papers, and I didn't know if we need to go into this now, but we'll certainly talk about it later. Do people know what orthologs and paralogs are? So what are they?
So if you have a gene and say it's copied within an organism and then passed down to that organism's descendants sort of in parallel so that each of the descendants has both copies of that gene, but then the genes sort of evolve independently of each other, semi-independently of each other, that's a paralog. And then an ortholog is when you have that gene, it has sort of a copy of it, but it's the organism's descendants each have a copy, and then those are independent of each other.
And so why is this of importance for phylogenetic studies?
Like sort of the pitfalls of it, or the like.
Oh,
I mean,
either.
you can have useful things from it, like rooting the tree of life was done with paralogs.
Right. What would be the problems that could be created by orthologs and paralogs?
You can't really tell if they actually are related to each other.
Right. So the main problem that a lot of phylogeneticists have worried about is if you're sampling organisms, and in particular if you don't have the whole genome of organisms, but even if you do, and you're lining up gene from organism 1 with gene from organism 2, you may actually be lining up paralogs. So if you wanted to build an evolutionary tree of mammals from globin sequences, you shouldn't take alpha globin from mouse with beta globin from humans with gamma globin from chimpanzees. You need to take alpha globin from each of them. And when you're going 2.5, 3 billion years back in time, it's entirely possible that every gene has undergone some type of duplication and weird paralogy, even ribosomal RNA genes. And therefore, you might get an evolutionary tree. The tree might be correct for those genes that you're looking at, but it might not represent what you think it does in terms of the species evolution. And so, for example, in the halophilic archaea, which we work on a little bit in my lab, and we've been sequencing genomes from this group, many of the halophilic archaea have multiple small subunit ribosomal RNA genes that are highly divergent from each other. And if you get the wrong ribosomal RNA and build a tree, as many people have done over history, you will misclassify these organisms because you're comparing, in essence, ribosomal RNA A with ribosomal RNA B between different organisms. And if you don't have the whole picture, you'll get an incorrect phylogeny.
So the whole things that they use to determine horizontal gene transfer or GC content, could you use that to try and make sure that you don't have ribosomal A and B from the same? If the organism has a certain GC content and another one doesn't, maybe be reflected.
The problem is that, let's say organism 1 has ribosomal RNA and ribosomal RNA B, and organism 2 has ribosomal RNA and ribosomal RNA B. And you just sequence one sequence from each of them, and you get B from one of them and A from the other. There's no simple way that anybody has figured out to tell B from A. There may be secondary structure elements or GC content elements that might allow you to tell them apart, but I don't know of any. So that's it.